Hello, dear students. I hope you're all staying home and keeping safe. Please continue to do so till the situation comes to a normal. Meanwhile, not wasting our time, let us take up the classes which we are missing. And I handle food microbiology for you, couple of units. And here we start the first unit of food microbiology. What is food microbiology? You might be wondering. What is there in food? What is there, the organisms, role of organisms in food? There are lots and lots of microorganisms on your food. The food we eat are rarely, if ever, sterile, I would say, because they carry microbial association whose composition depend upon which organism actually gains access to the particular food, and after gaining the access, how they grow, survive, and interact in the food over the time. Those organisms only are going to be isolated ultimately in the food. So, the microorganisms present will originate from the natural microflora of the raw material and those organisms introduced in the course of harvesting, processing, storage, and also in distribution. So, in this chapter, we are going to study actually what are all the factors which affect the growth, survival, and death of the microorganisms in each type of food. To start with, you all know the growth kinetics of your microorganisms, particularly the bacterial growth curve is what I have shown here. Looks like this. This is also called sigmoid growth curve. You're all very sure that they have four distinct phases: lag phase, exponential phase, stationary phase, and death phase. So what happens in lag phase is the cell number do not increase and the microorganisms have just entered to the atmosphere, that is a new food or whatever it may be, and it is getting adjusted to this new environment. Next exponential phase, you see that the numbers increase at the maximum rate with suitable conditions. So optimal growth happens there. Then the stationary phase due to the nutrition shortage, because it would have utilized all the nutrition and accumulation of waste products from the metabolism of the microorganisms. A few cell dies and a few cell multiply, keeping the living population stable. That is what we see in the stationary phase. Neither you can see the increase nor the decrease. Finally, you will encounter the death phase, where the population enters into the death phase, where the rate of cell death is higher than the rate of cell multiplication. Fine. So, these are all the four different phases you see, and your graph looks somewhat like a bent S. So, it is called sigmoid growth curve also. Okay. What we are going to study here is the shape of this growth curve is affected by various factors which is behind the scene. So what are those factors, whether they are inside the food, whether they are contaminated, whether they are from the atmosphere, and after entering from any sources, how it will act, what are the chemical composition of the food, how the chemical composition of the food in turn affects the growth of microorganisms. All these are going to be studied in detail. Okay. So, coming to the factors affecting the growth of microorganisms are four factors broadly classified as. One is intrinsic factors, extrinsic factors, implicit factors, and processing factors. Then, what are all these things? They're named as intrinsic, extrinsic, implicit, and processing. Which factor in particular are all coming under these broad headings? Let us see one by one. Intrinsic factors encompasses the pH, that is the hydrogen ion activity, then the water activity AW, redox potential, nutrient content, how rich is your food also matters, then antimicrobial substances present in the food, then the biological structures of the food. Coming to extrinsic, that is outside the food, I mean relative humidity, temperature, then gaseous atmosphere. Then, if I have dealt extrinsic factors now, you might be getting an doubt that why biological structures are not in extrinsic whereas it is an intrinsic. Because the biological structures are the outermost covering of the food itself. It doesn't make a part and parcel of the environment. Whereas relative humidity, temperature, gaseous atmosphere are environmental factors. That is why it is in intrinsic, not in extrinsic. Then coming to implicit factors. Implicit factors means within the food and the organism, what happens here, specific growth rate of the organism also depends. Suppose E. coli enters into the food, I cannot predict that this E. coli will be multiplying in this rate and it will reach the number this rate, because who knows, the food also may be harboring lactobacillus there. Lactobacillus may give in a fight with the E. coli there. So specific growth rate. Then antagonism, this is what fighting between the organisms. Antagonism also matters. Which organism outgrows which organism also is an important factor here. Commensalism, if the organisms are in the relationship of commensalism, that also matters in the number of the organisms in the food. Then synergism, together how well the friendship will be and how the effect will be brought about. So these are all the implicit factors which is solely dependent on the nature of the organism, not the nature of the food. Then comes the last, processing factors, wherein slicing, washing, packing, irradiation, pasteurization, whatever, however we handle the food, all this comes under the processing. So processing factors also. During this process also, there are a lot of chances where the organisms can get on to your food, and some changes can happen with the organism population also. So all these factors one by one we are going to deal in detail. One is the first one, the hydrogen ion concentration. What we have studied as hydrogen ion, pH means what? How do you define pH? pH is equal to the negative logarithm of the hydrogen ion activity. Please mind, it's negative logarithm, not the positive one. So the acidity or the alkalinity of the environment has a very profound effect on the activity and stability of macromolecules such as your enzymes. So the growth and metabolism of microorganisms are influenced by pH. You also agree, I also agree, because we set the pH while we prepare the media in the lab, right? So we don't have an habit of setting the pH same for all the media. You have been preparing the media, one for bacteria, one for fungi, one for yeast, all those things. So you go on setting different pH for different bacteria. Am I right? So generally you all know that bacteria 7 you will set, fungi somewhere 4.5 to 6 you will set, yeast much lower than that you will set. See, the function of the transportation of nutrients into the cell is also pH dependent. That's the reason we take care of setting the pH. You will be surprised to know even the morphology of some organisms can also be affected by pH. If the pH is not maintained properly, you will get a slight change in the morphology. I don't mean that the cocci becomes bacilli and the bacilli become cocci or vibrio or something like that. The long rods may appear short rods. The short